Hello, and welcome to today's HEAL Headliners webinar. My name is Carol Giannessi, and I will be assisting with the logistical support for this Zoom session. To provide comments or questions, please open the Q&A, which is located toward the bottom of your screen. If you need technical assistance, please type into the Q&A and one of our techs will respond. Finally, please be aware that today's session is being recorded. Closed captioning is available by clicking the CC icon at the bottom of your screen. I will now introduce today's moderator, Dr. D.P. Mohapatra, Program Director in Systems and Cognitive Neuroscience at the National Institute on Neurological Disorders and Stroke. D.P.?
Okay, thank you, Carol, and welcome everyone again to this webinar series. Really, we have exciting series going on, and this is another edition of this. So, as we all know, there is an urgent need for additional safe and effective treatment options for people who live with pain, opioid use disorders, or both. Yet the process of identifying molecules or processes that can serve as new treatment targets for pain often takes a long time. So today's presenters will describe how they are using very innovative approaches involving automation and machine learning to get results really faster for each of these conditions. So for our webinar today, our first speaker is Dr. Ismail Abdus Sabor. So Dr. Abdus Sabor is a Howard Hughes Medical Institute Freeman Hrabowski Scholar. I hope I pronounced it correctly. If not, my apologies. And he's also an associate professor of biological sciences at Columbia University. His research assesses how the brain differentially encodes responses to the sensation of touch, which is a very important element also in pain signaling. His work aims to further explain the perception of both pain as well as pleasure, as well as how each of these are altered in disease states or in different social contexts, which are extremely important in our quest towards developing new treatment therapeutics as well as treatment strategies. So with that, I'll give it to Dr. Abdus Sabor to go through your talk. So Ismail, please take it.
All right, good afternoon, everyone. Let me move this out of the way. All right. Good afternoon, everyone, and thanks, DP, for that kind introduction and the invitation to be here with you all. So over these next 15 minutes or so, I'll tell you about some of the approaches my lab has been taking to try to more robustly and accurately predict the animal's pain state. Now, just like to say, you know, all the work that I'll present today has definitely been supported by the NIH since I was a postdoc, and now more recently by the HEAL Initiative in my own research group. So here's a fundamental problem that we're trying to tackle: measuring pain in experimental animals. In the clinic with people, they can verbally articulate their underlying pain state, but for our experimental animals, the mice and rats can't talk to us, so this presents lots of challenges. So by and large, we've had to use behavior as a proxy to get at their internal pain state. And this has been quite challenging. So we've tried to improve a bit in this space. And I think, you know, in a quest to understand more about how pain works and to develop new and safe ways to treat pain, a fundamental prerequisite is that in our experimental animals, our preclinical models, we have to know how much pain an animal may be experiencing or even pain relief. So this is what we've been going after. So one approach, a starting point for some of our attempts, is to use high-speed videography, so to slow down really rapid movements. And I'll just say, as many in this audience are probably aware, a fundamental way to measure pain in rodents are reflexive assays where you apply a sensory stimulus and look for a response. But typically the response is how often an animal moved its paw, for example. But we think there's more that can be captured even in these rapid behaviors. So we've used high-speed videography. So let me just give you an example of how this looks. So in this first video, this is basically how things would look to the unaided eye if you were just scoring this live as an experimentalist. And we applied a noxious stimulus to this animal's paw, and you can see this animal clearly responded. So if you were using more traditional metrics, you would say, yes, clearly that animal responded. But just slowing down the same video to 2,000 frames per second, 2,000 snapshots per second, you see there's actually a lot more going on here. This animal actually jumped, shaking its paw and guarding it in this defensive sort of coping posture. You might have noticed some changes with its facial features. So there's a lot more information embedded, even within a few seconds of data, than just, yes, the animal moved its paw. And if we can extract that information in a meaningful way, perhaps this could help us, teach us a little bit more about accurately predicting how much pain this animal is in. So in our first version of this, we created this so-called mouse pain scale. This was based largely on the work I did as a postdoc with Professor Wen-Chin Liu at the University of Pennsylvania, where we apply either innocuous or noxious mechanical stimuli, and then by hand, we mapped and recorded and measured the most salient movement features to these different stimuli. Then we did some statistical modeling and machine learning, the take home of which is that we could make predictions on a trial by trial basis. This is what these individual dots are about the pain-like probability of a given response. One of the things I wanted to do when I opened my lab was to automate this platform, to speed it up and to make it a little bit more robust. So this work was led by the first two technicians in my lab who are all now off to graduate school. We again began by imaging the paw at really rapid speeds, but now instead of manual tracking, we use deep neural networks. And most frequently, we use algorithms called sleep and deep lab cut that are becoming mainstays in behavioral neuroscience, I would say. But then downstream of those track patterns, what do you do with that information? That presents another set of problems. So we generated our own software package that we call PAWS, which allows us to separate touch from pain stimuli, and then we can combine this into a single dimension if we'd like to put a number on pain. Let me show you a little bit about this, and then I'll move into newer stuff and unpublished work. So here's that same video, but now where we have the machine automatically tracking the paw through thousands of frames of videography. And if we do this, now I'm showing you work across multiple genetic strains. Here are just 6, but up until this point, we've tested 20 different inbred lines, 20 and counting. But here, if you look at the data I'm showing you here, you can see that we can separate out responses to these innocuous stimuli versus the noxious stimuli, just based off the nature of the paw withdrawal response. So here are the simple up-down lifts to the innocuous stimuli and these more sinusoidal elaborate patterns, often accompanied by jumping and grimace here with the face. So you can see is even, if we remove the animal and just looked at the nature of the response, we could probably predict the stimulus that this animal received. And I'll just say, without this high-speed imaging and this approach, these behaviors would look the same to the naked eye, just like an animal responded, okay? So we think the nature of the response could be an additional indicator for us to provide more information than just whether or not the animal moved its paw, because animals move their paws for all sorts of reasons. Sometimes it's not directly connected to the underlying pain state. All right, so here's our paw software, which stands for Pain Assessment at Withdrawal Speeds. And here are just some of the most salient features that we extract, like the height of the stimulated paw, the velocity in either an X or Y axis, how many things like paw shaking or paw guarding and the duration of those things can allow us to nicely separate out touch from pain. We've been able to show that we can take analogous sorts of approaches in rats with this high-speed videography and tracking these behaviors as a way to create a rat pain scale that works pretty well, just like we've been able to show in the mouse. And more recently, in the last month or so, we published this paper alongside colleagues Victoria Abrera and Ewan St. John Smith, and also Eric Ittrey, where we kind of put our heads together and we asked the question, what about using some of these modern tools to detect heightened pain states? There's a lot of information in this paper. If you're interested, take a look. I'll just highlight a little bit of it and mainly sort of some of our contributions here. So the heightened pain states we chose to go after are this carrageenan inflammatory pain model and this MIA, osteoarthritis knee joint model. And here are the time points we did our both electrophysiology and behavioral mapping based off some things in the literature and things that we uncovered. And what we can see, if I'll just show you some of the data with our PAWS platform, if we give an innocuous stimulus at baseline, again, the simple up-down lift. But if we look at this carrageenan model or this osteoarthritis knee joint model, and give an innocuous stimulus, you see the behaviors are a lot more exaggerated. The animal is having the shaking, guarding behavior, this defensive coping postures, and this is quantified here. This is the carrageenan model, and here the MIA model. See this mechanical allodynia here and hyperalgesia here. The point is, whatever the circuits are that are driving this defensive coping behavior from the DRG and spinal cord, but also the brain, are probably sensitized in these different models, okay? So even using this technology, we've been able to go back to models that people have been using for a really long time and still uncover new behavioral signatures, perhaps some of which could serve as sort of druggable behavioral targets. There's a lot more interesting, cool things in this paper. Again, check it out if you're interested. One of the things that I showed you, those 6 inbred genetic strains we've looked at, we've been extending this to look at a number of inbred strains. And this is part of the work that the HEAL initiative is funding for us. And with the goal of, perhaps if we find some mouse strains that have heightened pain sensitivity, then if we can get at the biological basis of that and maybe block it, that could be a way to get at a novel pain target. So let me just show you one strain that we've become quite interested in, this strain called SJL that came out of our screening using our behavioral platform. This strain would have been missed if we used traditional metrics because it mapped just like C57. But when we put it through our algorithms, we can detect these changes. So here's some ongoing unpublished work with this SJL strain. So you can see even at baseline, pre-injury, these animals have both innocuous or noxious stimuli, more of this guarding behavior. You see more shaking duration to a pinprick. And even if we look at the facial features of an animal, even to a cotton swab, the majority of these animals are displaying pain-like behaviors and to heavy pinprick, okay? Again, we would like to get at the biological basis of this. One of the things we're doing now is RNA sequencing in the DRG in an area in the brain called the BLA, basolateral amygdala, where we see heightened activity in these SJL mice when we do brain imaging, and we're starting to sequence there as well. Something else that came out of this, which is has us scratching our heads and might be interesting. We did some other phenotyping of these mice to see if there's anything else going on with them. So we did this anxiety-like assay that's commonly used in the field. We have an animal in the center of the arena. Animals that spend more time are considered less anxious versus those in the perimeter. And you see these animals almost never go to the center. So it looks like they also have an anxiety phenotype, even at baseline. Now, could their heightened pain be driving their anxiety, or anxiety driving pain, or both reinforcing one another? We don't know, but I think this is an interesting thing to kind of consider. And it also shows perhaps there could be interesting biology to uncover when we go beyond looking at the canonical C57 strain. All right, in my last few minutes as I wind down, I've told you a bit about our approaches on the pain assessment side, but what is left to improve with at least these mechanical assays? Well, as anyone knows in this audience, to do these mechanical, Vomfray or pinprick tests requires a highly skilled person, okay? And you want to, once that person is highly trained, you want to keep to that same person, because we all know that there's person-to-person variability with these manual application of these filaments. Requires someone to be able to have the physical capacity to do this, and it can just produce inconsistencies from trial to trial. So to kind of go after this question, and also the presence of a looming experimenter, it's also been reported to, mice and rats are prey animals, and you standing there on top of them, poking them, certainly probably has an effect on behavior of the animal, and that probably changes person to person. And some of that's already been reported from the Mogul lab. So to kind of circumvent this, we built this device we call the ARM, Automated Reproducible Mechanical Stimulator. And here's just a video. Here's our recording setup. And here's the device here on the left. So there's a camera here for allowing us to aim, and we can deliver innocuous or noxious stimuli to an animal's paw using this robot arm in a robust, reproducible fashion. This is new this is new days for us. I'll just show you a little bit of what we have as I wrap up. So one, it improves targeting accuracy. So if we take just researchers in the lab and have them manual here, a number of different researchers, and aim at a certain part of the paw versus the arm, you can see the researchers, even though they think they're doing a good job, that there's just quite a bit of spread that we don't see with the arm. I think this is important too. We can see with our high-speed imaging, and people in the audience may have seen this too. Sometimes you're down there, you're going to poke the animal, you think you hit their paw, but you hit the animal in the belly or something like that. They still respond, so maybe you check it as a response. But when we look at high-speed videography, it's striking to see how many times the manual experimenter is off from hitting the paw. And that's this evidence by this data here. Withdrawal latency. So this is something that we commonly see using like the Hargreaves apparatus, which is a nice proxy for pain sensation. With this device, because we can control when we turn it on and the animal's behavior, we can get nice withdrawal latencies. And you can see, we can separate out, there's a faster withdrawal latency with pinprick versus cotton swab. So now we can get the kinds of data with mechanical stimuli that we've traditionally only gotten with the Hargreaves thermal apparatus. The arm also, these are very new data, but the arm also is appearing to reduce some behavioral variability. If we look at some of our stimuli, how high the paw moved or the distance travel, we didn't detect statistical differences with the arm, but we do across, different manual experimenters. One thing you can see is that there's still some spread in the behavior with the arm. Giving precise stimulation does not remove all variability. There's still animal-to-animal variability, but it greatly reduces it compared to from person to person. One thing we also saw is that going back to Von Frey hair filaments, Von Freys are good because they have a calibrated force, but even delivering it with a person, there is some variability. So on the right, we're delivering 1, 2, or 6 grams Von Frey to a pressure arm. And you can see, even though it's the same filament, there's a little bit of variability from time to time, but this is largely lost when we deliver the same Von Frey with the arm. And then lastly, it allows remote work. So this is a student in the lab. She's not playing a video game. This is an Xbox controller, but we've coded it to be able to control the arm. And she's in a completely different room performing these assays. As you can see, here is our behavior room. There's a camera there, so we can aim. So the person could be at their desk, at home, as long as they have a Wi-Fi connection. And unsurprisingly, we're starting to see that the presence of a person in the room versus outside is drastically affecting the behavioral responses. But I think before we were unable to apply mechanical stimuli without a person in the room. So I think this adds another interesting degree of importance in these assays. All right, so just to wrap up, consistent, customizable stimuli, increased accessibility for people who may not be able-bodied, and it definitely speeds up experiments and cuts down variability. So with that, this is a relatively recent picture this summer from the lab. Thank all the amazing people who work in the lab, our funding over the years, especially the NIH HEAL initiative that's supporting all the work I discussed today. Thank you.
Okay, thank you, Dr. Abdul Sabur, for this really exciting and enlightening talk on advanced technology-based identification and assessment of pain behavioral signatures and rodent models of pain conditions. I'm sure there will be a lot of questions or interests, which we'll take afterwards in the Q&A session. So let's move to the second speaker. So our next speaker for today is Dr. Heike Wolf. So Dr. Wolf is a professor of pharmacology at the University of California, Davis. She is considered an expert on potassium channels. Her laboratory focuses mainly on the design of molecules to alter the functions of potassium channels to identify new research tools as well as potential drugs for treatment of multiple diseases, including pain, which has been her more recent focus. So her laboratory is particularly interested in those type of potassium channels that function within the cells in the immune system, in the cardiovascular system, as well as in the nervous systems, and specifically for our interest here in the pain systems. So I'd like to invite Dr. Wolf to take over and give a presentation. Over to you, Dr.
Thank you for this introduction. Let me see if I can share my screen here. Okay, here we are. Yeah, like DP said, this is going to be a more traditional drug discovery talk. We have a little R21 that is focused on KCA channel activators. This is a collaboration between my own lab at UC Davis and my former student, Hisong Shim, who is now at Lawrence Livermore. But let me start by explaining what we're using machine learning for. Virtual screening produces a lot of false positives that are not in agreement with crystal structures of ligand-protein complexes. So we're using AI for what is called post-classification. A post is the way a small molecule is sitting in a binding site in a protein. So we're using machine learning to differentiate the poses into those that look like something that would occur in a crystal structure and incorrect poses. So in many ways, it's like using AI to spot the bunny among all the cats, just that we're doing this in 3D. The problem we're trying to help with is opioid use disorder, which, as you know, affects millions of Americans. We're proposing a new target for substance use disorder, which are KCO2.2 potassium channels. These are calcium-activated potassium channels. They're found in neurons, but also expressed in the liver, urinary bladder, and arterial myocytes. These channels are calcium-activated. They're typically expressed close to their calcium sources, like, for example, the NMDA receptor. When intracellular calcium rises, it binds to calmodulin, which is constitutively associated with these channels, and then the channel opens. KCO2.2 channels, because they're voltage independent, can remain open at negative membrane potentials, and then they can mediate what is called after hyperpolarization, which regulates neuronal firing. So KCA2 channel activation will reduce neuronal excitability, while KCO2 channel inhibition will increase excitability. Loss of function mutations in KCO2.2 cause cerebellar ataxia, motor and language, and intellectual disability. KCO2.2 channels are best known for regulating learning and memory in the hippocampus. In the nucleus accumbens, KCO2.2 channels play a crucial role in drug seeking and drug craving during abstinence. There is data in both rodents and humans demonstrating that chronic alcohol use decreases KCO2.2 expression in nucleus accumbens neurons and increases excitability. There's also pharmacological validation for this target. Direct injection of apamine into the nucleus accumbens induces binge drinking in mice. In contrast, infusion of KCA2 activators like EBIO into the nucleus accumbens of rats or systemic treatment of mice with KCA2 activators reduces alcohol consumption and seeking. There's also data showing that morphine treatment in rats results in a similar reduction in KCO2.2 expression in nucleus accumbens neurons and increased action potential firing. So taken together, KCO2 channel activators constitute attractive novel therapeutics for preventing compulsive substance use. There might be some added benefits for KCO2 activators because they have also been shown to treat pain in rodent models and to treat PTSD. So they might have added benefits for the treatment of opioid-seeking behavior. So why not use the existing activators? You see them here, and the problem is that none of them are good enough. EBIO has low potency and lacks selectivity over KCS3.1. NS309 is potent, but lacks selectivity and has terrible PK. Our own SKA31 has good PK, but lacks selectivity. Our other compounds, SKA111 and 121, prefer the wrong channel, KCS3.1. CYPPA is KCO2.2 selective, but not potent. NS13001 is KCO2 selective and potent, but it lacks brain penetration. And then there is, of course, the issue of IP. If you really want to bring a compound into the clinic and have it benefit patients, you need to have a relatively recent patent. So our goal is to find a truly novel KCO2.2 activator. My laboratory has been working on the medicinal chemistry of KCA2 and the related KCA3.1 channels for close to 20 years. But we've always done this in the absence of structural information. Until recently, the McKinnon group published a cryo-EM structure of KCA3.1 in 2018. When the structure came out, we jumped on it. We used Rosetta ligand in combination with mutagenesis and electrophysiology to identify an activator binding pocket between the N lobe of calmodulin and the S4, S5 linker of the KCA channels. This work was done by my then graduate students, Hee-Sung Shim and Brendan Brown. And since we now had a structure, we started to do virtual screening, but we very quickly ran into the problem that I talked about at the beginning, that virtual screening identifies a lot of false positives. So my student Hee-Sung, who by that time was a postdoc and had a lot of at-home time in the early days of COVID, learned machine learning, and together with her husband, Hoo-Jin Kim at Lawrence Livermore, developed a post-classifier that was pretty good at classifying our KCS3.1 inhibitor data. We then applied for this heal.nih.gov, where we proposed to use a KCA2.2 homology model that was based on the KCS3.1 structure to perform virtual screening with GLIDE and Autodoc Wiener, followed by post-classification. And now I'm really going to get granular and talk about AIM-1, which was to validate our KCA2.2 model. We probed our model by running the FDA-approved library of 1,600 compounds through both pocket A and B in open state 1 and 2. And we had doped this library with 5 positive and 4 negative controls. My student Jerome Manera, who did this work, found that open state 2, with a very specific box size, correctly ranked all positive controls in the top 10 and did not rank any negative controls in the top 50. So we decided that these would be the perfect conditions for the larger screen. My other graduate student, Joshua Nussburg, validated our binding pocket through mutagenesis and electrophysiology and confirmed that our activators are really binding in pocket A in KCA2.2. Both GLIDE and Autodoc Wiener only identified frequent virtual screening hitters in the FDA library, like the beta blocker nebivolol, the antipsychotic haloperidol, and several highly polar antibiotics, which are pretty much the same compounds that everybody always finds in whatever virtual screen you do. So in summary, our AIM-1 results validated our KCS3.1 based homology model, but demonstrated that there currently really is no FDA-approved drug that could be repurposed for opioid use disorder, at least by targeting KCO2.2. So we proceeded to the larger screen. We ligand prepped and screened a ChemDiff library containing 130,000 ligands from 4 smaller focused libraries. We ran them with GLIDE through pocket A, B, and C, and through pockets A and B with Autodoc Wiener. But unfortunately, we found that the post classifier was absolutely not helpful. So we developed an alternative workflow where we're now taking the overlap, and this would be, for example, in pocket A, between glide and Autodoc Vena. We redock the overlap compounds with Rosetta ligand, which is computationally much more expensive and time-consuming. And we're basically using convergence in Rosetta ligand instead of the post classifier. We're also manually cherry-picking attractive compounds. So far, with the strategy, we have identified 2 micromolar activators. And I'm apologizing for not showing the structures here, but this webinar is a public disclosure. But of course, the big picture question is, why did our post classifier not work? Down here is a GitHub link for those who would like to try it. It is a machine learning model that classifies poses into those that are similar and dissimilar to what is seen in crystal structures. Like pretty much everybody in the field, we use the PDB-bind dataset to build our model. And this dataset is very, very biased towards structures of enzymes and enzyme inhibitors. These type of pick-and models work well when you apply them to proteins and ligands that are similar to what is found in PDB-bind. They don't work so well when you apply them to protein ligand complexes that are not represented in PDB-bind. And that is unfortunately the case for ion channels. When we made our model, there wasn't a single ion channel in the PDB-bind dataset. We had found an evaluation dataset of 40 ion channel ligand structures, which we had manually pulled out of PDB. But many of these are low resolution cryo-EM structures where you actually don't really see the ligand. So in many ways, this is like asking a model that was trained on cat and dog photos to identify giraffes. We are not the only people who are finding that AI is not or not yet helpful for identifying new small molecule ligands for new targets. Here's a paper in Frontiers in Pharmacology, Predicting or Pretending. Here's a preprint from a computational group at Oxford with a beautiful name, Postbusters. And here's another publication from a group at UC Berkeley saying exactly what we're seeing, that machine learned functions are not yet able to perform well when predicting new protein ligand complexes. We will, of course, continue to use our alternative workflow to look for novel KCO 2.2 activators. And with this, I would like to thank HEAL for the funding, you for your attention, and my students for their hard work. This work was done by Jerome Manera and Joshua Nussburg in my lab, and by Hisong Shim at Lawrence Livermore. Thank you.
Okay, thank you, Dr. Wolf, for such an enlightening talk and in-depth use of these models, which is really not much there before, and that to target the very fundamental excitability questions and how that can be utilized for developing therapeutics. That was really enlightening. Thank you again. So with that, we will open the Q&A session. So I believe we had some questions already. So yeah, I'd like to invite both Dr. Wolf and Dr. Abdus Sabor to have your microphone and video on. Yes, I see it. So yeah, the first question is for Dr. Abdus Sabor. So do you think the ARM can be adopted for any other animal models other than like rodents, even mice versus rats and other higher mammals or other animal models?
Yes, good question. You know, I sure hope so. I've spent some time talking to some veterinarians actually, and even our companion animals like dogs and cats, the problems that are raised today are also a problem there. How do you know your animal is in pain? And, they use sensory testing as well there, but I think some of the same limitations we have in rodents you find in companion animals, for example. So I'm a part of a new burgeoning collaboration with the investigator at Duke University, actually, Professor Kav Zarasa, where we're trying to move these tools over to dogs as a way to, see if we can recapitulate some of the signatures for pain, especially like with brain imaging, if the same sort of brain maps are there in both rodents and dogs. But also, I think with our companion animals, we have a chance to do some good there as well, not just basic science discovery, but that is a clinical problem, being able to objectively measure pain in our companion animals. So I think we are trying to move these tools into other animals. Yes. Early days.
And absolutely. And that could be really of high impact, and particularly the same theory, same one bullet for two targets. That would be really great. So the next question is for Dr. Wolf. So do you think the KCA2 channel, so desensitization or other forms of adaptation to multiple or any unique pharmacological activations?
Hey, that is a question that always comes up when you propose activators as drugs. So far, we've used KCA 2.3 and KCA 3.1 activators to look at endothelium-derived hyperpolarization. And so far, we haven't seen any desensitization there. KCA2 activators have also been used to treat ataxia relatively long term, and there hasn't been any desensitization. I know it's always a concern because with GABA you do see desensitization.
Yeah, that's correct.
But for example, with Kv7 activators, there doesn't seem to be desensitization. So I think it depends on the target if you see it.
Okay. And also, is the expression like of KCA2 is more wide in non-neuronal tissues, and how wide? Because that would be of real importance in terms of validating as well as moving further as target.
Okay, it is primarily expressed in neurons. Dense areas for KCA 2.2 are the hippocampus, the nucleus accumbens. There is expression in the liver, and nobody is quite sure what the channel actually does there. The knockouts have really no liver phenotype or anything like this. The loss of function mutations cause ataxia and learning disabilities. There is, of course, a concern that there is KCA 2.2 in the heart. It's actually being targeted with negative gating modulators to treat atrial fibrillation. But there's also SK1 and SK3. So I think the challenge is to make really selective activators that can differentiate between the KCA2 channels. Because until we have those, it's really hard to say what is the safety profile.
Absolutely. And within even neuronal populations in the peripheral nervous system, particularly in dorsal root or trigeminal, are there like cell type specific differences in the expression of these channels? Is anything known?
I believe so. But I don't think it has been really studied in a lot of detail. And again, this project really wasn't about treating pain. This was about opioid use disorder. Actually, when I wrote the grant, I wasn't even allowed to mention pain because it was all about treating substance use disorder.
Yep. No, I get it. I was just curious on whether
Yeah.
that could further be extended into analgesic therapeutic target,
There
quest
has
for
been
discovering. Yeah.
very little work. There is one group that has studied riluzole, which of course does multiple things. And part of its effect in pain models is mediated by KCL 2.2.
Mhm.
We've tested with the anticonvulsant screening program long ago a mixed compound that worked in formalin-induced pain, in sciatic nerve ligation, but that was a mixed sodium channel blocker KCL2 activator. So that was somewhat of a different beast, but that combination might actually be very useful for pain.
Yeah. So what are the next steps then for investigating the compounds that you already identified with your initial virtual skilling parameters?
The next steps will be to make them more potent and selective, because if we find a template, we really need to optimize it. You want to look at selectivity, and then when the compound is good enough, we would proceed to proof of concept studies.
Yeah, that absolutely sounds the way to go. So the next question is for Dr. Abdul Sabor. So can you tell us a bit more about the many different mouse lines that you have tested with automated system, and also do you have any plans to test outbred strains? I think you touched upon this in your talk, but a little bit more elaboration on that.
Yeah, sure. Yeah. So like I said, we've looked at about 20 inbred strains. When I was a postdoc, we did a little bit of work with CD1 and outbred strain. And I do agree. I think there is certainly value in looking at outbred strains, especially if we want to, sample the pain landscape and have our rodent results be applicable to human populations. Outbred strains make a lot of sense. And in rats, people mainly use outbred strains. And also, as I mentioned, Jeff Mogul and others are starting to show like outbred. People had traditionally only wanted to use inbred mice because they thought the data would be messier with outbred mouse strains, but it turns out maybe some of that was overhyped. But for some of these studies, we've been mainly using inbred mice because we do have a goal of maybe doing some QTL genetic mapping and trying to get at new pain targets. So I showed 6 of the strains we looked at. 10 of the strains we looked at are actually C57 sister strains. And we went after C57 sister strains with the idea, again, I showed C57 and SJL today, but if we want to get at genetic mapping, it could be easier to go for animals that have less genetic diversity. So there's one particular strain, I didn't show it today, called C57 Black 6 10S and J. And we've actually found a really amazing phenotype there where for some pain indications, they have 10 times more pain than C57 Black 6. So I think that's quite exciting, even at baseline. So if we can figure out like what is driving that high pain response at baseline, again, perhaps this could serve as another useful target. We have some interest in using the diversity outbred and collaborative cross strains, which NIH has been a part of supporting that. Those are on our to-do list as well to move in these platforms.
Yeah, in continuation with that, another question was whether you'll be able to use these with freely moving animals. That's, I'm pretty sure, is the next step you are already exploring.
Yes, that is very true. Our animals can move, but they are confined a bit to make our lives easier as experimentalists and at least recording the animals. But that is something that we are actively working on. We're also working on spontaneous behavior capture with like longitudinal imaging, 24/7 behavioral monitoring. I think a number of groups are starting to become interested in this, and we are as well. What is the animal doing when they're alone in their home cage, just, hanging out? Are there been animals in pain and chronic pain? Then perhaps there's something about their movements, their sleep-wake patterns, things of that nature that can be detected if you were able to record that animal unconstrained 24/7. So this is a data management nightmare, but I think there are solutions that are coming online, and this is another thing that we are trying to get going now.
Yeah, thank you. So the next question, it's a question plus comment for Dr. Wolf. So your search for FDA-approved drugs turned up negative, but your negative results are so important. So can you speak to the value of publishing negative data in protein structure, especially related to drug development for unmet medical need, including the substance use disorder and pain? So this more kind of like a comment and a big picture,
Okay.
which we always hear. These are really important.
I'm a big fan of publishing negative data. That, of course, requires journals that allow you space and supplements where you can publish this data. I should probably disclose I'm chief field editor for Frontiers in Pharmacology, which is a journal that does take negative data. So we will definitely publish this, probably as part of a larger paper. Because again, if it's only negative data by itself, people typically don't want to read it. But when it's part of something else, you can package it.
Yeah, it's always a challenge question. So another question for you would, will there be any effort to determine the structure of ligand channel complexes that you're now probing?
Of course, not with an R21, but actually one of my collaborators has a structure of KCO 2.2, which he will be showing at Biophysics and at the Ion Channel meeting in Irvine in 2 weeks. So I hope this will speed it up. It's always difficult to get small molecules into structures. You might have the channel without a small molecule in it, then you soak the small molecule into the structure. And then because of symmetry issues in cryo-EM, you might see that the structure moved, that the selectivity filter changed, but it's sometimes still hard to find the small molecule in the structure. But of course, there are ongoing efforts for it.
Yeah, thank you. So the next question is for Dr. Abdul Sabor. So it's more of a question as well as a request. So your data provides an eye-opening new look on old models. So how can you and your tools and findings be socialized broadly to potentially reopen investigation once that are closed? So of course, I know you are already doing a lot of effort on outreaching and sharing these models, and but I would let you elaborate a little more on that.
Yeah, we sure are. I think the strength of tools are getting the tools in people's hands, and for us as a community to kind of use these tools. And maybe that's a little plug for myself and some colleagues. There's a course next week at the Jackson Labs where we're taking biologists and teaching them how to use a lot of these machine learning tools to study pain and other sorts of behavioral tools. And I think the questioner asked something really good, and that comes up to us a lot. Is it time to kind of reevaluate some things that we had overlooked in the past with a fresh set of eyes? I think that's a really good idea. One thing that we are gearing up to do is to take a list of some of the analgesics that has showed promise in rodents, but maybe fell in the clinic, good positive hits and negative hits, and to see how those things would look in our hands. Could we have predicted that maybe some of these targets weren't the best? It would be great if this platform could be used as a screening tool to add confidence to which targets we should move forward with in the clinic. So I think looking at some old targets and some new ones and kind of doing a pharmacologic screen with these tools is something that we definitely want to do.
Absolutely. I mean, this is an important step. It's just more in the beginning, but as more and more people utilize, that will really not only expand the use of it, but also be very vital for developing.
Right. I think there's this whole emerging field that people are calling computational neuroethology. And I guess some of the stuff I presented is just a small part of it, but there's a whole large ecosystem. So I think for pain, for anxiety, for depression, you name it, I think there's just been a renaissance in tools and better behavioral phenotyping. So the degree to which we can kind of infuse these tools into our research programs, I think all of our research will improve it, particularly pain research as well. Yeah.
Absolutely. Thank you. So we still have a couple of questions. One for Dr. Wolf. So how much further do you think the virtual screening process can be refined to identify promising compounds even more accurately?
That really depends on, like every AI algorithm, the more data you give it, the better it gets. So virtual screening is working relatively well for enzymes where there is a lot of data to train on. For ion channels, we're really just missing data to train on.
Mhm.
Um,
That's true.
which doesn't mean that you shouldn't try it, because structure-based drug design is intellectually so much more satisfying than just blind screening.
Yeah, that's true. More and more structure and more structural structure determination with bound drugs that are agonist, antagonist to really expand that much more. So I have a question for both of you to address. So where do you see actually the greatest potential for automation or machine learning in addressing the pain and opioid crisis in the future? It's a very big open-ended question, but we really want to pick your brain and know your thoughts on this.
Can I go second?
Okay, Dr. Wolf, you go first.
I think Ishmael's model was a perfect example of how you can automate image analysis, how you can pull so much more out of images than just saying like the mouse withdrew its paw. I think this is a perfect example of machine learning. And programming precise movements. I mean, if you use machine learning to poke a mouse in its foot or wash the windows in a high-rise building with a drone, it's very useful technology.
Right. Yeah. And just continuing on that, I mean, obviously, I guess I'm a bit biased, but, I do think moving to this place where we can not go in with so much experimenter, investigator bias on what we think behaviors mean to an animal, but allow the animal's behavior to kind of speak to us. And I think there's like a whole vast array of behaviors that we just haven't been scoring or, for simplicity and taking more reductionist approaches. But I think these tools allow us to take an agnostic approach. And, I'm quite excited about where the future, I think there's so much more we can get out of our experimental animals than we have in the past. So I'm excited for the future of these tools. And I think, training, you and I have talked about this, DP, training, I think is going to be really important for the next generation because I mean, I'm not so old, but I'm still in that generation where, just as a biologist, you may be a little bit afraid of computation or coding and doing some of these sorts of things. But I think, today's upcoming researchers want to be competent and moving between experiment and computation and not having that fear. And to the degree that people are able to do that, I think that would just open up so much for us.
Absolutely, I do agree with that. Like early training and more multidisciplinary training and really moving trainees through a completely different area for like bench scientists to go and learn and use these automation and also inviting folks from coding, the modeling, computational science world to come to the collaborate with bench scientists and develop and really validate these models, which can predict a lot. And of course, linking these models in future to maybe data like heavy bio and psychosocial database models taken from human data, and then how to really combine these two types of models to come up with much better predictive value for therapeutics development as well as effective management, how it can directly inform and predict the treatment outcomes for these opioid use disorders, pain, as well as many other disorders.
Agree.
Yeah, and maybe you could also come to some more standardized measures for clinical trials.
Absolutely. That's also another critical point, which is very often missed. Let me check if there are any other questions. So this point, I did not see any other questions are there. It was really, really wonderful talk by both of you and a lot of discussion. I would like to thank you again, Drs. Abdul Sabur and Wolf, for such a great lightning talks as well as so engaging discussion. So this is just, I believe, just the beginning. We'll be discussing way more and more in depth on this area, and we'll continue with that. So thanks again. So with that, I'll pass it back to Carol. I think you are muted, Carol.
Yep, thank you. I'm here with a plug for our next webinar, which I can just share my screen. Oh, nope, I don't need to. All right, so our next webinar will be November 3rd from 2 to 3, and the theme is healing across the lifespan. So I really hope you can join us for that. And thank you for attending today. It's been a really great discussion.
Okay, thank you, everyone.
Thank you, everyone.
Thanks, bye.
Thank you.